Now that we've actually done a principal component analysis, we want to plot it and understand the principal components in the context of the metadata we collected for our samples, that information that's stored in our study design file that we read in as targets. And so to do that, let's just kind of return to this idea of what PRComp gives back to us. When we ran our principal component analysis, PRComp produces a result that is stored as a list data type. We've talked about what a list is, and we even kind of detailed a few of the elements in this list, and I've provided comments here to kind of help you as you work through this. But one particular element in this list, X, is really going to be what we'll draw on for making our PCA plot. And the reason we'll focus on X and not other parts of this list is that X contains information about how much each sample really influenced each principal component, and we'll call that loadings. So if I highlight PCA res $X and I evaluate that, I see that what I'm really dealing with is a data matrix where each row is a sample and each column is a principal component. And importantly, the data has both magnitude and direction. So if you think back to that silly example I gave with height and weight for a hypothetical principal component analysis plot, I highlighted, once I drew PC1, I highlighted points, samples, people that were at one extreme of PC1 and samples or data points at the other end of PC1. So you can see how that relates to our data, right? We have samples that weigh very negatively, so at one extreme end of PC1, and samples that weigh very positively, so at the other end of that PC1. And so this data is really no different really than the height and weight data that I showed you. We should be able to appreciate, we will be able to appreciate how much each sample contributed to each principal component and to visualize that in a simple scatter plot. So to do that, we will turn back to our friend ggplot2, and to be able to take this data matrix and plot it in ggplot2, we immediately have a sort of prerequisite, and I've discussed this before, and that's that ggplot2 and all the other packages in the tidyverse only play nice with data frames, and this is a data matrix. So first order of business is to use as Tibble from the Tibble package to convert this X element in PCA res to a data frame, and in particular a Tibble. So we'll run this line of code, and that produces a nice data frame version of the matrix I just showed you. We'll now plot this, and then what I'd like to do is actually we'll plot it first before we talk about the code. We'll use the appearance of the plot to then come back to this and revisit this code and kind of iterate over how we might change this plot to make it not just aesthetically more pleasing, but actually more informative. So as we run this chunk over this block of ggplot code, we quickly get a PCA plot, and this should look pretty standard to you. It's an XY plot with each sample represented as a point here in some location in this 2D space. And even just looking at this and thinking about what you've learned about ggplot2 from the last lecture, you should be able to kind of sketch out how this might look given ggplot's layered grammar of graphics, right? We know that we have a data frame. That's the first argument in that ggplot function. So we give it this data frame. We know we have aesthetics we need to map. PC1 maps to the x-axis, PC2 to the y. We know we would suspect this is a geom point function that's run in ggplot2 to produce these round points to position them on this coordinate space. And we have some theme that gives it the kind of aesthetics of the box with the faded grid bars. So now let's return to the code, and indeed we see ggplots working on this data frame, of course. We have some aesthetic mappings, PC1 to X, PC2 to Y, and although we can't appreciate it in the plot, we have also mapped our sample labels, which are just a character vector of simple sample names. We've mapped that to an aesthetic called label that we can call on later to actually throw labels on these samples, and we'll do that in just a minute. We were right that this is a geom point plot. We dictated how large we want those points to be, and then we've added some labels. Labels to the top of the plot, a title, label to the bottom, a caption. and then X and Y axis labels. And this is just one thing to highlight here is that we can actually use not just character information or text to label our axes, but we can also pull from objects we created earlier in our script to label the axes. And this becomes really useful for creating axes that are both informative and accurate. So for example, PC1 as an axis label for the x-axis is not useful here. We wouldn't know if you only had PC1 listed what proportion of the variance is explained by that principal component, and that's a key part of a PCA analysis. So we want to include the word PC1, but with the percent variance explained. Same thing with PC2. So we could do that manually, right? We know from our analysis above what proportion of variance is explained by PC1. We could just kind of add that in manually, but we could maybe do this in a smarter way by actually calling on that PC per object that we created, which is a numeric vector of the percent variance explained by all 10 principal components, and then subset out just the element we want to display in the axis. In this case, just the first element in that numeric vector, which is the percent variance explained by PC1. So we're using the paste0 function here to do all this, and that's just a variation on the base paste function. The 0 here means, and I think I've discussed this before, means 0 separators between the things you'll paste together. And the things you'll paste together are separated by commas in the expression. So PC1, open parenthesis, then paste that together with the first element in this numeric vector. And then with no spaces, again, paste0, put that together with a parenthesis, a percentage sign, and then again, no spaces, paste0, put that together with a closed parenthesis. Now, there's no reason why I needed to have this out here by itself. I could have just put that in here, since that would effectively accomplish the same thing. But nevertheless, paste0 will stick all these things together, and so it will take this and generate it for you, which is great, because it means that no matter what you change up here in your principal component analysis, if you rerun and reproduce the plot, the axes will always be accurate. They'll always reflect the correct percent variance based on the most recent PCA analysis. Okay, so that's how we get our X and Y labels, and then we applied a theme, of course, to end the graphic, right? So those are our layers of our ggplot. Okay. Now I told you, now that we have this graphic, let's refine this and iterate on this a bit. The first thing I would say is that a PCA, a principal component analysis, is an unsupervised dimensional reduction method. Unsupervised here means that the PCA has no knowledge of our group assignments or variables in our study, right? When we ran the PCA, it didn't ask for, are these samples from control or from disease. It had no knowledge of that, but we have knowledge of that. So we can layer over information about our study onto this unsupervised analysis. So how do we do that? Well, it's actually really, really easy. So we have a variable that we created earlier called group, and that group is just healthy versus disease. So I'm going to actually add that to my aesthetics. It's that easy. I'm going to say color equals group. And remember, group is just a factor where it indicates each sample in the set of 10, whether it's healthy or disease, and then it respects that that's a factor, and there's really two levels in that character vector. So if I replot now with this color mapping in the aesthetics, now I get something that's actually really informative. I see that all the points over here are from healthy participants. All the points over here are from individuals that had the disease. And so since that difference spans across this x-axis, I now can see that PC1, which accounts for almost 70% of the variation in my data, really is telling me about disease status, healthy versus disease. I now have attached meaning to PC1, and the only way I did that was by layering over information from my study design into this graphic. All right, so that takes care of PC1. I now understand what PC1 is, and that's a big relief and really exciting. in part because I can explain what PC1 is. So I don't have to worry about it being something maybe I don't understand, one of Donald Rumsfeld's unknown unknowns. But the other reason it's exciting is that because, my phenotype of interest, disease status here, is contributing to the most variation in my data, it means that downstream, when I do a differential gene testing, I'll find lots of genes different between healthy and disease, because that aspect of my data is really driving most of the variation. So this bodes very well for the steps we'll do downstream. PC2, it's not separating disease from healthy. It's really separating this one data point from all the others. And so this is an example of where a principal component can tell you not about a covariate in your data, but about an outlier. So how would we find out which sample that is? Well, we kind of have an inkling as to what it is. We've seen it as an outlier in the hierarchical clustering dendrogram that we produced. But we could also take advantage of the fact that we have added our sample labels and map them to this label aesthetic. And so we can use the geome label as another layer in our plot and replot this. And now what we'll get is still colored, but now instead of points, we've colored over those points with a geome label. Remember, these labels or these layers stack on top of each other. So we actually covered over our data points here. We could move them with an offset over to the right, over to the left, up or down, so that both the geome point layer below it and the geome label were visible, but we're not worried about that. We can see that right away, that healthy sample number 5, the one that got only a 1/10 to 1/20th of the number of reads as these other samples, is really consistently different than all my other healthy samples. All right, what else can we do here? I'm going to just comment that back out and replot that. So now I understand what PC1 is. I understand what PC2 is. I've now accounted for 80% of the variance in my data just with those first two principal components. Right, we replot it back to where we were before. And I thought I'd just highlight two other things. One is it's usually a mistake to present your PCA plot where the axes are drawn in a way that they suggest equal scale when your labels suggest very different proportion or percent variance explained by those principal components. In other words, if this is 70% of the variance, it should probably represent a longer axis than the axis that represents 11% of the variance. And so we can fix that very easily by changing, by adding in a coordinate fixed function from ggplot. So just to highlight here, because of the way RStudio is graphing, if I make my graph wider, it will make it scale, right? But that's not how the graphic would save if I exported it. Conversely, if I drag it in, it will make it perfectly square. And again, that's not how it will save it. So I can fix the coordinates, and I'm just going to take that comment out and rerun this ggplot. And regardless of how I have my window sized here, it will keep the coordinates fixed to reflect the data on those axes. So now this plot, no matter how I slide this out, will always keep those coordinates fixed based on the scale of the data. Okay, so that's useful, especially if you're going to publish, to make sure those axes are scaled to reflect the variance they explain. The last thing I'll just highlight is you can use the stat ellipse function to apply a statistical ellipse around data, and that ellipse will recognize grouping variables as well. So if you have two groupings on your plot, it will attempt to draw two ellipses. So if we take a look at what this looks like, we end up with now a PCA plot where it's drawn these data ellipses around our samples. And so if they really are distinct, you should get nice tight ellipses that give you some confidence that what you see by eye really is true. Okay, so that's the essence of making a principal component analysis plot. I posed some questions for you down here to sort of think about what would this PCA plot look like if you used raw counts, the stuff we stored in my counts, instead of log2 counts per million. What are the disadvantages of looking at a PCA plot using a simple 2D representation? And so think a little bit about this type of graph that we've made here. Okay, so I want to talk a bit about this last point. What are the disadvantages of looking at a PCA result using a simple XY plot? And to think about that a bit more, let's head back to our slides and let's just remember what we've generated. So, really our PCA analysis was pretty simple. From the scree plot and from our actual plot of the loadings of each sample on the first two principal components, we can see that PC1 explains most of the variance, and we can see that disease status is easily identified as PC1, while this single outlier accounts for PC2. But what if it's not so simple? I mean, many experiments have lots and lots of covariates to explore, and sometimes there's substantial variation explained by multiple principal components. Maybe PC1 explains 50% of the variance in the data, but PC2 still explains 45%, right? And so in that case, understanding some of these successive principal components and how they relate to your study is critical. And so that's where you can really spend time painting different covariates and information about your study onto these PCA plots. And it can get a little tiresome to do that if you're just looking at PC1 versus PC2, and then perhaps changing this to PC2 versus PC3 and putting on different, then going back through these painting on different covariates. So one of the things I would recommend is taking advantage of something, a type of graphing that's been around for a very long time, in fact, very long time, called a small multiples. And the concept of small multiples is really simple. You produce similar graphs using the same axes and labels. And this allows you to make the graph itself very small and make many of them because the axes, the labels are all the same. There's only one thing changing. This is also sometimes called faceting, but for a principal component analysis, this becomes a really useful way to explore the relationship between your sample metadata and the principal components. So I love this very old 1800s data from census data about deaths and deaths by age across different states in the U.S. And this is a classic example of small multiples where the y-axis is the age range from just born to 80 years plus. And then the x-axis is magnitude or number of deaths. So further out is more deaths. And then the multiples, the small multiples here are the states. That's your faceting. Same graph type, same scales, just faceted by state. And so change this to principal components and you basically have what I'm proposing can be a useful way to explore principal components. And before I leave this, what is definitely my favorite example of small multiples, I'll just highlight that the census from the late 1800s, I would not want to live in Montana, Idaho, or Dakota and be a male. So male are the shaded plots. And again, as we climb up, we're going up in age. And you see there's a heck of a lot of young men dying in their 20s and 30s. And it's probably because in Dakota, Idaho, and Montana, a lot of the work was very dangerous, mining, logging, things that were involved substantial risk. And that resulted in high mortality for men that primarily occupied those jobs. Also note that there's extremely high death early in age here, of course, from measles, just to highlight the impact that vaccines have had on reducing early life mortality. Okay, so great. So then how do we sort of think about making a small multiples graph? And I'm just going to go back to our studio here. And that takes us to our next code chunk, which is producing this small multiples plot. So the way we're going to do that is still relying on PCA res $x, but we don't need small multiples for all 10 principal components. We know that we account for 80% of the variance just in the first two principal components, but we'll go a couple extra because that third and fourth principal component account for a couple percent of the variance as well. So that means instead of just using PCA res $x, I just want the first four columns of that. So let's look at PCA res $x so we recall. I just want the first four columns here. And so because this is a data matrix, because it's rectangular, whether it's a data matrix or a data frame, I can subset using that convenient subsetting operation that I discussed before, where I can choose rows on the left side of a comma or columns on the right side. And so I don't want to subset rows, I just want to subset columns. And I can provide a range or select certain columns, continuous or discontinuous. I want columns 1 through 4. So this will return just columns 1 through 4 and produce this data matrix. And so that's what I'm going to operate on. But now we have to talk about this, the first time we've encountered this pipe function, which is here. So this is something extremely useful. And so we need to just take one slide to talk about what it is and why we'll be using it basically all the time from this point out. So we're going to use this pipe function to streamline our code. And I'm going to give you an example of what I mean by that. So let's take a hypothetical bit of code where we have some data, we'll call my data. We're going to do something to it with some function. And the result of that's going to be stored in called my new object. All right. We then need to do something else to that, right? Because we don't just do one thing in our code. So now we're operating on my new object and we're using some other function, do something else. So we're doing something else to my new object, and that creates something called my new object. This process carries on and on. Now we're doing something different to my new object to make my new 3 object. You get the idea. The point is, every time we're storing these intermediate objects, we're creating a lot of bloat in our code. And so we have to look at this code and really recognize the essential elements, that there is really data, there's 3 functions, do something else, do something different, and then there's really this object that we're creating from doing these things. So what the Magrittr package will allow us to do is actually access a function that's really just an operator. And so that is the percent greater than percent. It's really the main, there's sort of slight variations on this pipe function within the Magrittr package, but it is really what this package does is provide this functionality of a pipe operator. And it's part of the tidyverse, but it's just loaded automatically when you load any tidyverse package. So you don't really see it as sort of in the background. And so that'll allow us to take something like this and reduce it to this, right? Which is we can pipe everything and sort of cascade it down through the code. So we can start off by just saying, here's my data. Let's pipe that forward to the functions below. Okay, instead of having to put data in each of these parentheses. We can then, once we've piped the data to the next line, and again, this has a layered feel to it. Once we've piped our data to the next line to do something to it, we don't need to dictate what we're doing it to. We've already piped the data to that function. And whatever do something produces will not be stored as an object as it was here. It'll be piped down to the next function. So there's no storing intermediate objects, which really reduces, I think the busyness of the code. So it makes it more readable. Same thing, after we do something else to that object, we don't store that result. It gets piped to the next line, and then we do something different. And now the culmination of all these steps is stored as a single object. So these intermediate objects are not saved in any way. Okay, so that's all going to be accomplished with this pipe function in Magrittr. And so the way I suggest you using this is to start by always taking your data that you want to use and piping that down to the next line and the result of that to the next line, etc. And all of this piping gets assigned to a single object. All right, so this subsetted data matrix that I just showed you gets piped down to convert it into a tibble, because again, we want to do stuff to it in the tidyverse, and so that means it needs to be a data frame. And then the thing we're going to do to it is really simple. We're going to use the add column function to add two columns, one called sample and one called group. And sample is going to be our sample labels, and group is going to be our group assignment, healthy or disease. All right, so we'll run this bit of code, and we'll take a look at what we produced, and we can see that indeed it was turned into a tibble, and two columns were added, one that has the sample ID and one that has the group assignment. Now that I have these in, I'm going to pivot this data, and we talked about this idea of pivot longer from the tidier package and how it'll take wide, messy data and make it long, thus the name pivot longer, long, tidy data. And we discussed what tidy data really meant, that each variable forms a column and each observation forms a row. And so if we look at the data we have here, immediately see that it's not tidy. We have a variable called principal component spread across multiple columns instead of being in one column. So the pivot longer function will take this data frame. It'll capture columns, the columns labeled PC1 through, that's the colon, through PC4. And it'll take these column names and store them in a new variable called PC, a new column of our tidy data frame. And the values for that shown under those four columns, these numeric data, will be stored in a new column called loadings. All right, if we run this bit of code and we take a look at PCA pivot, now we have a tidy version of the data frame I just showed you. We have now, instead of this wide data, where our variable principal component is spread across multiple columns, we have a single column for principal component and a single column for the numeric data. And I described to you why this is valuable. It's because now we can access the numeric data as a single numeric vector, and that makes it really easy to plot and then facet or bin by aspects in the other columns, other variables, right? The group, the principal component, the sample. Recall that we used the same sort of display in the previous lecture as well. Okay, so now that we've pivoted this data, we're ready to plot it using ggplot, where the X will be our samples, the Y will be our loadings, and we'll fill the color by group. We'll use a geometric bar. We will, first time we've done this, we'll facet wrap, and we'll facet wrap, in other words, this is the actual small multiples part of things. We will produce multiple facets of this graph based on the information stored in the PC variable here, so by PC. And then we've got some labels, a theme applied, and then I flip the coordinates to make the graph display in a way that's a bit more readable. All right, so let's just, enough talking, let's run this and see what we produce. Okay, so I got an error, and I'm not sure why. Let's see here if I clear my graphics window, and I rerun this. And now we get the plot. So I just had an issue with my graphics window. It always helps to clear it. And now I can see that I see a loadings plot. Remember, magnitude and direction in that X element of the PCA result plotted for each principal component as a sort of faceted small multiples graph. And I've colored this by the group variable. I filled by group, because that really makes it very apparent that, as we already saw from the initial graph, PC1 is really telling us about healthy versus disease. PC2 is telling us about this one outlier, HSO5, that sticks out one direction, so heavy loading on principal component 2. PC3 and PC4, I don't know what they are, right? They don't match with disease and healthy. They aren't pointing towards a single outlier. PC4 has this unusual back and forth pattern where 2 of the healthy samples are loading in one direction on PC4, and 2 disease samples are loading in the same direction, while the others are weighted on PC4 in the other way. This might point to a situation where you'd want to look and see if these 2 healthy and these 2 disease were part of the same batch. and then these other samples were part of another batch. So that would be a batch effect contributing to PC4. PC4 is only a few percentage points of the variation, but nonetheless, that could represent a batch effect. Okay, so I think that's a good point to just head back and recall that that's really the value in being able to pipe these functions and work quickly with our data. So my word of advice to you is to take the time to thoroughly explore your data before downstream analysis. There are examples, many in the literature, of where this kind of data exploration has not been as rigorously pursued, and it's resulted in real issues. So I'll highlight a couple of those for you. One is it specifically relates to PCA analysis, but another that just relates to how we encode and think about metadata. And so this is a PNAS paper by Michael Snyder's group, a very well-established, very successful lab that is really a pioneering lab for RNA-seq. So, issues with multivariate analysis can happen to anybody. And this is a PCA result shown in their paper, where they have all the human samples clustering together and all the mouse samples clustering together. And this is data from a large, ENCODE RNA-seq data set where they looked at various tissues in human and mouse. And what they describe is this distinct clustering. It's really sort of the basis for the fundamental finding in their paper. And they say that to date, various studies have found similarities between human and mice at molecular level. Indeed, mice are a big model for humans. And they highlight that in general, differences dominate similarities between the two species. These findings provide the basis for understanding differences in phenotypes and responses to conditions in humans and mice. Basically, what they're saying is, wow, isn't it surprising that human and mice cluster so differently? We would expect that brain should cluster together and lung should cluster together, because we know that there are conserved transcriptional programs that make brain what brain is, and similarly what make lung do what lung tissue does. So we would expect first clustering by tissue, and then within each tissue a separation by species. But in fact, that wasn't what they observed. They observed clustering dominated by a species effect. And so this caused some concern, and that concern was articulated in a paper on Faculty of 1000 by Yoav Galad's group. And they say, the implications of this observation that human and mouse gene expression may be clustering by species more than by tissues could be profound. To a large degree, modern biology is built upon this idea of homologous gene regulatory networks driving tissue development and conservation of tissue function, etc. I'm paraphrasing here. If true, if the results of that PNAS paper are true, this challenges these observations and the biological basis of homology. And from a more practical perspective, how do we think about mice as a model then, if they really are so distinct transcriptionally from human? And so, this would call into question the utility of the mouse and perhaps other non-human animals as a useful model system for biomedical research. And so, to really start to understand the results from that PNAS paper, Galad's group, really started to dig into the data that was analyzed in the paper from the Snyder lab. And I'll just remind you that FASTQ files have a lot of information preserved in this header. And we didn't really talk much about what's in the header, but there can be a lot of information encoded in that header. In fact, here's an example of one from the paper in question, and you can actually identify everything from the machine run to the flow cell to the lane on the flow cell that was used to the exact region or tile of the lane and the X and Y position in that tile, which read it is, forward or reverse, and what the barcode on that read is. So a lot of information stored in there, and you can use that if you're savvy and can parse these FASTQ files and pull that information out. You can basically parse the samples out. And what Galad's group did was they went through these samples, whether they were human or mouse, and they asked, well, were they, what else could explain the variation observed on that PCA plot other than just species? And so they were able to segregate these samples based on whether they were run on different machines or different flow cells or different lanes. And what you can see is a big batching effect here where the human samples were largely run on one machine, on one run, on one flow cell in 2 different lanes, while all the mouse samples were run on different machines, different runs, different flow cells, and different lanes. And so this really raises the question of whether that separation they observed on the PCA plot was truly due to species, or was it due to one of the other confounding variables, which was they were totally different machines, different runs, etc. And so that played out in the literature where they reanalyzed the data and showed that if you sort of remove this species variable, there's really no way to discern now a separation, because again, species is confounded with other aspects of the study design that weren't really well interrogated. And this played out in the literature in terms of questions raised about, well, how do we really interpret this then if those variables are confounded? And we don't need to go over that. Another example that I'll just highlight is a more recent one from the end of 2019 about a retraction of a paper that talked about the impact of certain interventions on outcomes for patients with COPD. And they wrote this was into JAMA, so a major journal for the medical profession. We write to report a programming error. And they say, the identified programming error was in a file used for preparation of the analytic data sets and occurred while the variable referred to as one of their group assignments was randomly assigned a format of 1 to 2 and had been reformatted to a 0 to 1. And so essentially, they changed the encoding of their variable. You could think of this as if you encoded sex as 1 and 2 instead of male and female, and then you switch to 0 and 1 at some point, you might accidentally switch which of these encoded. Now the 1, instead of encoding for male, encodes for female. So this is the kind of issues that can arise when we're not really careful about our metadata. Okay, so back to the code. The last things we want to cover today really deal with analysis of our data frame and doing that using dplyr. And we're going to pick up on that in another part of the lecture.